I'm continuing this is part 2 of chapter 8. Um, so cells do manage energy resources through what we call energy coupling. Energy coupling means that um, when you use hexagonic process to drive endergonic reaction, so it means in essence that hexagonic is going to release energy into the environment, and that energy released into the environment is now used for another process. Does that make sense? So think about it now. Energy coupling will be a view as photosynthesis that happen in plants, is going to produce oxygen for us. We breathe in that oxygen, we use the glucose in the product and use it to drive our own life's activities. We use it to perform other mechanical activities. So that's energy coupling, when the product of an hexagonic reaction is used to drive an endergonic reaction. Now, most energy coupling in cells are obviously mediated by ATP. All right. So what is ATP? ATP is adenosine triphosphate. Adenosine triphosphate. Adenosine triphosphate means that there is one adenine, which you are seeing here, and there are 3 phosphate attached to it. All right. So it's made up of, I'm going to annotate this. So it's made up of a ribose sugar, which is what you are seeing here. This is the sugar. All right. Then it's made up of 3 nitrogenous bases. I mean, 3 phosphates. You see, these are the 3 phosphates attached to it, and 1 adenine, which is right there, adenine or adenine. We use A as a symbol. So that's where A triphosphate came into place. Now, in addition to energy coupling, ATP function as one of the nucleoside phosphates that are used to make RNA as well. Let's go. Now, hydrolysis of ATP simply means breaking down of ATP. Hydrolysis means addition of water to break down the bond. So when you have ATP, if you look at this ATP, for instance, now, like I told you, there are 3 phosphates attached to a sugar molecule and adenine, which is a nitrogenous base. Now, for ATP powers are chemical reactions, right? So we say that if there's going to be any chemical reaction in our system, metabolism, ATP powers a reaction. What ATP actually does is to give energy to protein and for protein to do the action. Protein is the machine that does the work. ATP is the gas right now. But how does ATP power chemical reaction? This is it actually. Now, I told you there are 3 phosphates here. Now, the way ATP power chemical reaction is that this phosphate here, which is the last phosphate here, will be removed. When this phosphate, it's actually what gives energy to a protein. So this phosphate is going to be released and is going to bind to a protein or to the enzyme that should perform the action. And that is what we call energy. It is like you have $300 with you, 100, 100, and 100. Then someone needs to do an activity. But for the person to be able to do the activity, the person needs $100. So you give out $100, then you have $200 left. That is actually what ATP does. So the way ATP actually power any reaction is to release one phosphate. When it releases one phosphate, it's now going to become ADP. This phosphate then is what gives energy to the substance that needs to be powered. Now, so ATP releases more energy with a lot of phosphate than most other molecules could deliver. Repulsion between the negative charges of the 3 phosphate groups is what creates the potential energy. Now, the triphosphate tail is the chemical equivalent of compressed spring. Right, let's go. Now, cellular work, either mechanical, transport, and chemical, is powered by ATP hydrolysis, ATP broken down. In the cell, energy from hexagonic hydrolysis of ATP, now that's an hexagonic reaction because it's releasing energy into the environment. So in essence, what I'm saying is this. When you have ATP broken down into ADP, right, plus phosphate, this is an hexagonic reaction because it's giving out energy to the environment. So this hydrolysis reaction is an hexagonic reaction, all right? And this now will be used to power the energy released here will be used to power another reaction. That is what we call energy coupling. Now, in essence now, this is what we are talking about. So you see, I told you that the phosphate, the terminal phosphate released from the ATP is going to bind to a protein to catalyze a reaction, to cause a reaction to happen. So look at this reaction now. You have glutamic acid here, and what is going to happen? The ATP is going to release phosphate, which is going to be here. This is the ATP now. You see, it's going to release a phosphate. The phosphate released from this ATP is what you see here. So it has become glutamine now that has been phosphorylated. Phosphate has been added to it. Does that make sense? Now, when the phosphate is added to it, then it powers the reaction, and then the glutamine will now react with NH3, and then reaction happen. All right. This means the energy required for this reaction to happen is what the ATP has come to give. All right. And now the glutamine is produced. All right. In essence, if you look at this, you will notice that the energy required here to perform this reaction is plus 34 by this itself. Now, when ATP is added, ATP requirement, the energy that ATP brings is negative 3.3, it's negative 7.3 kilowatts. By the time you check the delta G of this and the delta G of this and the delta G of this, you see that the answer turned out to be negative 3.9. This is negative, means that the reaction is spontaneous. Again, I just want to point to you the spontaneity and non-spontaneity of a reaction. Now, look at this, transport and mechanical work. For this to be done, again, ATP needs to be broken down. ATP needs to be hydrolyzed. Now, see, when ATP is hydrolyzed, phosphate is going to be added to this protein. When this protein receives phosphate, it's going to open. And then it's going to carry molecules that it should carry. And then you see it opens up here again and transports it. That is because ATP is available. Again, the same thing here. For muscle or any other activities to be done, for movement of molecules to happen, you see ATP needs to bind. When ATP phosphorylates it, then the cell or the vesicle is able to move. So ATP is involved in all of these activities. Now, how will ATP be regenerated? Now, I told you that when ATP lose one of its phosphates to the environment or to power a reaction, what it's going to now remain ADP, which will be in the system. So ADP is generated by addition of another phosphate to the ADP that was also there before. So if I want to regenerate, so I had this reaction before, right? I had ATP that splitted to give me ADP plus a phosphate, which is already used up. This is already used up, not available again. Now, if I want to get my ATP back, how can I get ATP back? The way to get ATP back is simply to look for another phosphate and add it to the ADP, and then it's going to give me ADP, ATP again. Now, so ATP is regenerated by just getting another phosphate that will be added to the ATP, and it will become ATP. Don't forget, ADP means adenosine diphosphate, and ATP is adenosine triphosphate. Now, free energy needed to phosphorylate ADP is going to come from hexagonic breakdown of another product, like another reaction. Now, the shortening of inorganic phosphate and energy is called ATP cycle. As it moves between ADP to ATP, ADP to ATP, is called organic cycle. It couples energy yielding processes to energy consuming processes. So let's go. So that is now, how do you get the phosphates that can be added to ADP to make it ATP? Food sources, right? Now, enzymes speed up metabolic reactions by lowering the barrier. Now, in essence, what we are saying is this: for any reaction to take place, there's an energy that is required. Just like this, for any transaction to hope to happen in a store, there is a particular amount of money you have to take to the store to purchase something. If you go to a store and you don't have enough money to buy what you want to buy, then there will be no transaction. Let's say you want to buy a Krog, for instance. Now, a Krog averagely costs about $50. For you to buy the Crocs, you need to take $50 to the store to buy the Crocs. That is the energy that is required for that transaction. So the same way, for every transaction you want to make in this body, there is an energy requirement for those transactions. However, there is a way to reduce the energy required. Now, the same way, like I use the Crocs, if you want to buy a Crocs now, you go to Macy's, it costs about $50, right? However, sometimes there may be so like now, sometimes there could be, let's say, President's Day that just passed now. They are going to say we are doing 40% off. And 40% off will automatically take about $20 off the $50. And now you can purchase a Croc for $30. Now, that discount that they gave you is what we call enzyme. What has the enzyme does? The enzyme has reduced the energy required for that purchase. The money required for that purchase has now reduced from $50 to $30. Now, let's assume that you want to buy a Crocs and you have planned that you were going to buy it in April when you'll have saved some money. But now, President's sale came up and you saw that, okay, you need to wait till April to save $50. But now, President's Day reduced the price of Crocs to $30. So, and you already have $30. You were going to wait till April, May, March, and April to save $50, but now you have $30. So you are simply going to go ahead and buy the Crocs. What has the discount does for you? The discounts made it possible for you to buy the Crocs earlier than you had planned to buy it because it required less money. The same thing enzyme does to reaction. It helps a reaction to go faster because the energy requirement has been reduced. So spontaneous reactions do not need added energy, but they can be slow enough to be imperceptible. For example, the hydrolysis of sucrose to glucose and fructose is spontaneous. At room temperature, a solution of sucrose in sterile water will sit for years without appreciable hydrolysis. Now let's go. Now, what does catalyst do? Catalyst will reduce the energy required. The price of the Crocs are reduced by the enzyme. Like discount, the discount is the energy or the catalyst. Now, a catalyst is a chemical agent that speeds up chemical reaction without being consumed in the reaction. Now, an enzyme is a macromolecule typically that acts as a catalyst to speed up chemical specific reaction. Now, for example, now, adding the enzyme sucrase to a sucrose solution at room temperature will catalyze the reaction within seconds instead of waiting for years. So instead of this reaction to wait for years to break down sucrose into glucose and fructose, and that will sit for years, because it doesn't need energy for it to happen. It can happen without energy, but it's going to take a long time. So to speed up the chemical reaction, to buy your Crocs without waiting till April, you need a discount. So you need an enzyme to make a chemical reaction to be faster. And how does it make it faster? By lowering the energy required for the reaction. So this is what we call the energy required for a reaction is what we call activation barrier or activation energy. So the activation energy is the energy that is required for a reaction to proceed. That is what we call activation energy. Now, every energy, a chemical reactions between molecules involve bonds, breaking, and bond forming. So before a reaction can happen, it's either that you are breaking down a large molecule, which will break the bond, or you are building up a small molecule, which will build bond. So you are either building bond or breaking bond. You are either doing a catabolic reaction or an anabolic reaction. A molecule must be contorted into a highly unstable state is what we call entropy. And I talk about that in the part 1. Before bonds can break to start reaction. To reach this stage, the molecule must absorb energy. This is what we call activation energy. Before you can buy your Crocs, you must save a lot of money. That is what we call activation energy. Now, the initial energy needed to break the bond of a reaction is called activation energy. You see that? So the activation energy is the energy required to break the bond. All right. Heat in the form of thermal energy absorbed from the surrounding often supplies this activation energy. That's why we say a reaction will go faster when there is heat. Molecules become unstable when enough energy is absorbed to break the bond. This is called transition state. Now you know that even if you want to make food, you have to put the food on the stove, and then the food have to be applied to the food. If there's no heat, the food will not be cooked. The food will just be soaked. All right? Now, as atoms settle into new, more stable bond, energy is released into the environment. In an hexagonic reaction, the formation of new bonds will release more energy than what was invested in the environment. That is what we call hexagonic reaction. If it were entagonic, more energy will be invested than what will be released. All right, so hexagonic reaction will release more energy into the environment. Now, this is an analogy of what I've been explaining. Again, for any chemical reaction to proceed, an energy barrier must be overcome. The energy barrier that must be overcome is what we call activation energy. All right, now, for the reaction to happen, activation energy must be overcome. Now, in this case now, we say that delta G is less than 0. That means this reaction is a spontaneous reaction. Now, the energy required to cause this reaction to happen is right here. So let's say this is about 50 kilojoules, which is the energy required. So 50 kJ. This must be met before the reaction can proceed. If this is not met, the reaction is going to stay there. Now, this is the reactant side, and this is the energy barrier that must be achieved. Now, but by the time this reaction was completed, you see there is more energy released into the product. So the activation energy provides a barrier that determines the rate of spontaneous reaction, right? For some reaction, activation energy. So EA means activation energy. EA, this means activation energy. Is low enough that the thermal energy at room temperature is sufficient to overcome the activation energy. Most reactions have high activation energy, and we need additional energy, usually heat, to reach the transition state. All right, that is that. Now, adding heat is not useful. It's not a useful way to speed reactions in cell because it can cause problem to proteins, right? It can denature the protein. So that is the reason why enzyme becomes necessary for protein. It is also impractical because it will speed up all reaction, not just the one that are needed. So instead, organism will carry out catalysis which is the process by which a catalyst selectively speed up a chemical reaction without itself being consumed. Now, this is what we are talking about in terms of enzyme catalyzing reaction. So if you look at this now, let me use my annotation. Give me a moment. Now, if you look at this, for this reaction to happen by itself without an enzyme, that is the black line here, this is what you need. This means the energy required here is this much. It is like you need $50 to purchase your Crocs. You will need to wait till April to gather this money. But there was President's Day sales, and then they lowered the price to $30. They did 40% off, and that brought it to $30. Now with $30, you are able to go ahead and purchase your Crocs. Now, what has happened is that the activation energy has reduced. It has reduced. You see, it has reduced from 50 to 30 because of the enzyme. Here, this requires higher activation energy. This requires lower activation energy. So what enzyme does is to lower the activation energy for a reaction to take place. So an enzyme catalyzes a reaction by lowering, by lowering the activation energy barrier enough for the reactions to occur, even without increasing the temperature beyond normal. Now, an enzyme cannot change delta G. It can only speed up a reaction that will eventually occur anyways. So enzyme doesn't change Gibbs free energy like I told you earlier. Enzyme will only reduce the energy required for the reaction to proceed. Now, this is what we call specificity of an enzyme. It means an enzyme is specific to the substrate or the reactant it can catalyze. So I tell people, like now, when you say you have lactose intolerance, but you have maltase in your system, you have sucrase, you have amylase, you have a lot of enzyme peptidase. All of those enzymes are there, but they can't go ahead and break down lactose because lactose can only be broken down by the lactase enzyme. That is specificity of an enzyme. And why is that important? Now, even though both of these, so now I have different, I'm going to, let's see, I should have different markers here. Now, both of these are markers. Now, both of these are markers, but even though they are the same they are markers, but the cover for this is not going to fit into this. It's not going to fit into it. Does that make, do you see that? It's not going to fit into it because they are not the same. So that's the same way enzymes are specific with the substrate or the reactant they affect. Reactant is the same thing as substrate. Substrate is from the word substance, which is also reactant. So they cannot catalyze, an enzyme cannot catalyze all reactions. Why? They must fit, there must be fitness between the enzyme and the reactant. So it must be like a door, a key and a lock. The key must fit into the lock for the key to open the door. So for an enzyme to be able to unlock a reaction, there must be fitness. All right. So you see that this is, for instance, now, all right. So for instance, now you have this as the enzyme, at the substrate. Here's a substrate here. Here's a substrate. For this substrate to fit into this reaction, it must be able to fit exactly into the enzyme. You see, it must fit straight. Now, when it fits in straight, then it makes the enzyme substrate complex. All right. Now, if that doesn't happen, the reaction cannot be supported by the enzyme. Now, so most enzymes are named by here. So you see enzyme, you see sucrase, lactase, all of those are the names. That anytime you see ASE, except when you see it in people's name, that is an enzyme. All right, now look at this. Now on this, you see the substrate. Here's the substrate. binding to the active site on the enzyme. So why we say that enzymes are specific with their action is that the enzyme have an active site. The active site only allow specific shape of a substrate. If the substrate cannot fit into the active site, the enzyme cannot catalyze the reaction. So lactose will be able to fit into lactase, and that's why it is only lactase that can break down lactose. Now that is what we call, you see, induced fit. So this substrate must be able to fit into this enzyme, and then the enzyme can close up with it. Now, how is this important in enzyme reaction, in breaking down chemical reaction? Now, or in speeding up chemical reaction to lower the activation energy. Now, I told you that what we call chemical reaction is that there is either you are building a bond or you are breaking a bond. So in cases when a bond needs to be broken, for instance, now, what is the relationship between enzyme and the substrate? When this substrate fits into this place and the enzyme binds to it, you know what happened? If it's a catabolic reaction where we need this substance to be broken down into two, 1 and 2, the force of the enzyme in binding to the substrate, having a strong grip on the substrate, we weaken the bond that surrounds the enzyme, that surround the substrate. The bond that join the substrate together will be weakened by the pressure of the enzyme on the substrate. So if they are not induced fit, that will not be possible. So the pressure that the enzyme brings holds the substrate will weaken the bond. And now the energy required to break that bond is already reduced because the enzyme has weakened the bond. If it's a case when it's an anabolic reaction, then the substrate will be able to bring, the enzyme will be able to bring the substrate together. And I will show you that as we go. All right. So the substrate is typically held in the enzyme active site by weak bonds, such as hydrogen bonds. The conversion of the substrate to product happens rapidly, but product is released from the active site. Because enzymes emerge from reactions in their original form, some small amount can cause huge metabolic activity. So you see, look at this. So substrate enter the active site. Here's the substrate in the active site. They are held together, and then the active site will lower EA by bringing them together. So bringing them together is a key factor. Bringing them together is a key factor in lowering the activation energy. All right. Then the substrate are now converted into product, and these are the product. Then the product are released. Then the active site is again available for another substrate to bind. Then the cycle continues. Again, substrates, enzymes are not used up in chemical reaction. They only help the chemical reaction. They are never going to be part of the product. So if you see this, you see that the enzyme remains intact. The enzyme is not part of the product. These are new products that are not part of the enzymes at all. So enzymes use a variety of mechanisms to lower EA. Number one, the substrate may be oriented to facilitate the reaction. So it may be to position the substrate very well. Substrate is the same thing as reactant, like I told you. Substrate may be stretched to make the bonds easier to break. You see that? Weakening the bond. The active site may provide microenvironment that favors the reaction. So like temperature, the environment may become conducive. All right. Amino acid in the active site may participate in the reaction as well. So rate of enzyme catalyzed reaction can be sped up by increasing the substrate concentration. That's number one. When all enzyme molecules have their active site engaged, the enzyme will be saturated. If the enzyme is saturated, the reaction can then be sped up by adding more enzymes. All right. Now, enzyme activity can be affected by general environmental factors such as temperature. I told you earlier that temperature affects enzyme activity. pH also affects enzyme activity. All right. So the way that enzyme speed up chemical reaction could be by increasing, if you increase the substrate, enzyme activity can also be increased. temperature, I told you earlier that if you look at their protein, their enzymes have optimum temperature. There are some temperature where they are the best. So some of you that are currently doing the lab, you see that sometimes you may have done the experiment when you use catalase and potato for an experiment, and you were trying to check at what pH, at what pH is this, at what you use that to check substrate concentration. Then sometimes you use different temperature to check the catalyst to see what temperature will produce the best results. So there is a temperature that will favor the enzyme more and will make the enzyme to produce better result than you. If you remember, you were checking bubbles. You see the one that made higher bubbles at that temperature. If the temperature have higher bubbles, then that means that temperature is the most effective for that enzyme. So those are different temperatures. So we call the temperature that is going to produce the best result for an enzyme, we call it optimum temperature. Then we call the pH optimum pH. Now, each enzyme has an optimal temperature at which it can catalyze reaction, all right? The optimum temperature does not mean highest temperature and it does not mean lowest temperature. Optimum temperature simply means it's usually a little above the middle, but it does not mean the middle. All right. So up to this point, the reaction rate will increase with increasing temperature. Beyond this point, the rate will also drop. So the rate where the enzyme is going to produce the best result is what we call optimum temperature. Again, it does not mean average temperature. It's usually above average, but it's not the lowest and it's not the highest. Enzyme begin to denature at temperature that is beyond the optimum as well. Now, optimum temperature of an enzyme is dependent on the environment in which the enzyme typically functions. For example, the optimum temperature for human enzyme is about 37 degrees, whereas the optimum temperature for thermophilic bacteria is actually 75 degrees. So it depends on the enzyme, it depends on the environment based on the organism that is of concern. Each enzyme has an optimum temperature at which it catalyzes its reaction at the highest possible rates. Up to this point, the reaction rate is going to increase with increasing temperature. And when it goes beyond that, the enzyme activity will drop. And then we say that the protein or the enzyme has been denatured. So looking at this now, here is the optimum temperature for this protein enzyme. Here's the optimum temperature. You see, right after that, the enzyme begins to drop. So for human now, this is about 37 degrees. For bacteria is about 75 degrees. Again, look at this pH, the same concept, optimum pH here. Anything below that, the protein begins to denature, begins to die or break down, which is what we call denaturation. Now, there are some things we call coenzyme. Coenzyme or cofactors rather, they are non-protein helper that bind to the enzyme permanently or reversibly with the substrate. So we have some enzymes the cofactors that we call inorganic cofactors. Those are metal atoms such as zinc, iron, copper. Those are inorganic cofactors. But they are organic cofactors. We call them coenzymes. Now, most vitamins are coenzymes. What's the function of coenzymes? They help enzymes to do its job. So in cases where enzymes have some little lapses that they need help, they come to support the enzyme. They don't work in place of enzyme. They help to support the enzyme. All right. Now, those are coenzymes. That's vitamins. That's why they advise us to eat vitamins. I'll be talking about that now. So certain chemical selectively inhibit the action of specific enzymes. Now, if an inhibitor forms covalent bonds with the enzyme, then the inhibition is usually irreversible. I'll talk about it. Many inhibitors bind to the enzyme by weak interaction, which results in irreversible inhibition. Now, look at this. We have an inhibitor that we call competitive inhibitor. I told you that substrate binds to the active site on the enzyme. So when you locate the active site on the enzyme, this, for instance, is the substrate, and the active site is where it's going to bind. But there's another inhibitor which also look like a substrate that also want to bind to that area. Now, whoever binds first determines what is going to happen. So if this inhibitor bind to the substrate, to the active site, then the substrate will not be able to bind. What that means is that reaction will not be able to happen. And that is part of the way that reactions are regulated. So competitive inhibitors closely resemble the substrate, and they can bind to the active site. Enzyme productivity is reduced because the inhibitor blocks the substrate from entering the active site. So increasing substrate concentration can overcome this drive. So if there's a lot of substrate, then the inhibitor will not be able to bind. But if the substrate delays, inhibitor binds, that means the reaction cannot happen. Now, there's another kind of inhibitors that does not compete with the substrate. So we call that non-competitive inhibitor. Now, it's going to bind the inhibitor is going to bind to another location on the substrate. And here is it, we call that location, we call it the allosteric site. We don't call it active site. Here is what we call allosteric site. Now, what is going to happen? The moment the inhibitor binds, once the inhibitor binds there, it will change the shape of the active site from this. So if the substrate now binds to the change shape, there will be no reaction. So this is it. Once the inhibitor binds here to the allosteric site, allosteric is spelled allosteric. So this site is called allosteric. I'll type it right here. Allosteric site. So once the inhibitor binds to the allosteric site, then the shape, so here is the allosteric site. All right, the shape of the so once the inhibitor binds to the allosteric site, the shape of the active site is going to change, just like what you've seen here. The shape has changed. Now, if the substrate now binds, there will be no reaction. It doesn't matter. The fact that the shape has changed, it will change the shape. Once the shape is changed, there cannot be any reaction, even if the substrate binds. Now, toxins and poisons are often irreversible enzyme inhibitors. For example, sarin gas was used in a chemical attack in Syria in 2017, killing and injuring hundreds. Sarin will bind covalently to the active site of acetylcholine esterase, which is an enzyme which is involved in nervous system coordination. So once sarin binds to the acetylcholine esterase, then that means there won't be acetylcholine. Acetylcholine is the neurotransmitter that helps to transmit the message from the neuron to the muscles. So if acetylcholine esterase helps to break down acetylcholine that is there. So if there's no acetylcholine to break down the acetylcholine, there's no acetylcholine esterase to break down the acetylcholine, that means the muscle action potential keeps firing, and that's a lot of trouble. So that will cause a problem in the action of the nervous system. It's a kind of inhibitor that binds to the acetylcholinesterase, which cause a problem. Other examples include pesticide and antibiotic. Enzymes are proteins encoded by genes, and changes in gene mutation will lead to changes in the amino acid composition of the enzyme. And if the amino acid is altered, particularly in the active site, that can result in novel enzyme activity or altered substrate specificity. Now, if mutation results in new enzyme function, this in cases when it is beneficial, like when you talk about somebody is resistant, somebody does not have a kind of allergic reaction that some people have, for instance. No, for instance, repeated mutation and selective selection of beta galactosidase enzyme in E. coli resulted in change in sugar substrate under the lab condition. So these are changes that happen. Though there are some mutations that we call beneficial mutation, there are some that are not beneficial, but all mutations are not necessarily bad. So the regulation of enzyme activity is what I've talked about earlier. Chemical chaos will result if cells' metabolic pathway are operating simultaneously. So cell can regulate metabolic pathway by switching on and off the gene that encode for specific enzymes, or by regulating the activities of the enzyme based on need. All right, so this is what I was pointing earlier. Allosteric regulation occurs when a regulatory molecule binds to a protein at one site and affects the protein function at another site. This type of regulation may either inhibit or stimulate activity. Most allosterically regulated enzymes are made from polypeptide subunit with its own active site. I'll talk about it. The complex oscillates between the two shapes, one catalytically active and the other inactive. So this is what I was talking about. So allosteric enzymes allosteric regulators bind to the allosteric site on the enzyme, and that will prevent the substrate from binding. It will affect this enzyme activity. So allosteric activator and inhibitor. So allosteric enzymes with 4 subunits. So these are allosteric enzyme. It has 4 subunits. There's a active site. There's a regulatory site. So once the substrate is supposed to bind to the active site. And so you see the substrate binds to the active site there. Now, for here, you see the activator binding there, supporting the substrate to bind. When the activator binds, it supports the substrate to bind. But in the other case here, once the inhibitor, which is the red, binds, you see the shape of the active site will change and will not allow the substrate to bind. In that case, the substrate will not be able to bind and the enzyme becomes inactive. Now, cooperativity, another type of allosteric enzyme, you see, in cooperativity, the substrate binding to one active site triggers a shape change in the enzyme that stabilizes the active form for all other sites. All right, so you see, once this bind, the other active site will open normally, unlike here, and then the substrate will be able to bind. So that's what we call it, substitute. Feedback inhibition. This is actually okay. Feedback inhibition is, in feedback inhibition, the end product of a metabolic pathway shuts down the pathway. So feedback is invariably, what it's saying is that if there is no need for a chemical reaction to happen, then the product, the brain tells that, okay, now there's no need for this reaction to happen because there's no need again. And so that's going to stop the process. That's what we call feedback. All right. So feedback inhibition prevents a cell from wasting chemical resources by synthesizing more product than is needed. All right. So in case of this, isoleucine is used up here by the cell. All right. Feedback inhibition says isoleucine binds to allosteric site, and it's saying, yeah, there's no need to make anything again. And so the process is going to stop. But in cases when there's available site, the threonine is going to process and then the whole process continues. But the concept of feedback is saying we already have enough. So there's no need to make any other thing. So isoleucine is going to bind to the allosteric site, and then there will be nothing to bind again because active site is saying I'm no longer available. All right. So that's it for this chapter. Please watch the chapter. If you have questions, reach out to me. Thank you.